best part. So so let me start by saying thanks to Paul for the invitation and for organizing this webinar. And it's a pleasure to be here in my home, seeing you through the computer. Well, it's not a real pleasure, but it is what it is, isn't it? Let me start by saying that what I'm trying to convey this talk is the complexity of actually reconstructing networks from different kind of data. I'm using different kind of approaches and indices. The interesting part of this is that people have been doing this for a while already and publishing a lot of papers on infer kind of networks, which, as we will show here, are very difficult to reconstruct. And that's really a kind of an open problem as yet. In terms of how you can infer the graph, which is the graph that actually represents the architecture of ecological interactions and the environment where these interactions are taking place. So it's a very complex problem. So as a kind of introduction, let's see, how do I? Okay, as a kind of introduction, just to make sure you will understand some of the jargon I will be using in this talk. What we are trying to infer are interactions, and you have different kind of interactions, some of which are, we call it trophic, which is related to something eating something else, and that's a very direct interaction. You have a predator that eats a prey, like here you can see a crab eating a mussel. Okay, so you have also different kind of non-trophic interactions that I will call NTI here. You have non-trophic interactions that are positive, for example, substrates or mussel beds that provide shelter for organisms. Like here, you see the mussel, which actually is a substrate and shelter for different kind of organisms on its top. You can also have a kind of behavioral changes that associated with that create a better kind of a possibility to escape predation, for example, just gathering together, or you also have also non-trophic interaction associated to competition for space or resources. Okay, so all these are kind of interactions that are going on in an ecological networks. So you have positive, you have negative, you have direct associated to trophic interactions, and also you have indirect interactions. You know, and also in addition to that, you have an environmental gradients through space. So I'm putting here a picture of the intertidal because that's where I will talk in part of my talk will be focused on this. And the intertidal, basically you can divide it in three main regions, which is going from the bottom up is the subtidal, is the area which is also always covered by water. Then you have the middle intertidal zone, which is a kind of fluctuating. I mean, on high tide it's covered with water, but in low tide it's not. And then you have the high intertidal zone, which is not covered by water. So, but it's very close to the water. So you get all the spray, all the waves and so on. So you have a gradient, an environmental gradient. So, so the question here is that it looks like things can be together in the same place and interact, or they can be together in the same place and do not interact. They are there just because they like the place, like they like to be covered by water all the time, or they like this the kind of middle zone and so forth. So it's kind of a complicated picture because you have environmental influences, you have species interactions, and you also have the species responding to climate, as we know that species has been responding to climate change in the marine realm. So what we do is we try to reconstruct a network. So we have a network to begin with. So that's the beauty of this work, is that we do have a network, and I'm not going to talk too much about this because Sergio Navarrete is going to talk about the network itself. So we have a network where we know the trophic, the non-trophic, and then the positive non-trophic and the negative non-trophic. So so we have this template, and so we can use all the kind of data to reconstruct this to see how well we do in reconstructing this complexity. And what kind of data is the most common that people use? It is co-occurrence data in terms of presence and absence of species on the space. So suppose that you go and sample, in this case, we sample like 70 sites or 49 sites, I think. And what you have, you record in each site, each sampling unit is like a small quadrant. So you record things together in this quadrant, and you move also from site to site along a gradient of a couple 100 kilometers along the coast, and you can record which species are together with which other species, and you can have species A here and B sharing a lot of sites together, or few, or kind of overlapping in terms of one being present with almost always that the other is present. So you can use a covariance or calculate correlations on these present absences, and eventually construct a species by species kind of interaction based on this co-occurrence and covariance. So you say, okay, all the significant correlations or covariances will be I will take it as interactions, and then you can kind of reconstruct your network. So that's what basically what is usually done, either with co-occurrence or with abundance data. So so we tried that, and just to see if this method actually was strong enough to allow us to make some sensible inferences about the complexity of the network. So we have this number of sites here at different times, you can see, but also a different point in space. So you have 49 sites in total, and so we applied this co-occurrence analysis to these networks, and basically what we obtain is that well, the co-occurrence are not analogous to classical ecological networks, by the way. So the positive interaction are the ones that are most detectable at all, and mostly there is a strong kind of of environmental signal in terms of species co-occurring because they are in the same environment, not because they are interacting. Okay, so what is interesting though is that the sensitivity, which is also called the true positive rate, so is is actually the probability of detecting a link which is present, increase as you increase the occurrence of the number of sites where a species was present. You see, but as the species are more widely distributed, or you have more occurrences, you are able to detect true interactions, but at the same time, a lot of your specificity, which is the true negative, decreases. So you start detecting a lot of false negatives interactions. So these two things are correlated inversely. So it creates a big problem of inference, because if you want to do the right thing, there's a point where you start doing the right and the wrong at the same time. So that doesn't actually get you too far on this. And it looks like what is really more important is where you're located across the gradient in the intertidal, more than what you are interacting with. So here you see that detected links are very good for negative trophic interactions, and but in terms of proportions, or the one that you detect the most are the positive non-trophic interactions, and they change between kind of environments or positions along the intertidal. And in fact, you can see that here is the exercise. What we plot here is what you reconstruct, the kind of links that you reconstruct if you just use high tide samples, or if you use, let me use my high tide samples, or low tide samples, or middle tide samples. So you reconstruct different colors, and the red ones are negative trophic interactions, and the blue ones are positive. So depending from where the samples come, you get a different reality of a network. So that's a very strong influence of the place where you're looking at. And also there's a lot of weak interactions that seems to be very important in actually affecting the network, and most of them may not be well recovered by this kind of covariance or correlation methods. Also, the true positive rate of habitat forming species. These habitat forming species are species that then by itself they provide habitat for other species, where some other species can recruit. For example, the larvae can settle down there. So these kind of habitat forming species have a very high effect size, which is a measure of the correlation that they observe. So they are highly correlated, the same as some of the small grazers that are highly connected to many other kind of species. So it looks like there's many different factors that affect the kind of correlation that you get, that might not necessarily be associated with interactions, but it just is more associated with the kind of things you do and where you live, mostly. So that wasn't a very kind of successful story in terms of inferring the network using this method that people really use. So we strongly kind of caution against this kind of methodology. There is a paper on this. If anyone is interested, we can pass it around. So just to make sure that this, or just in order to control a little bit more for this, we actually decided to go into more into a theoretical analysis, and we created our own networks, just to make sure. So we created network using this simple stochastic equation here for with a predator prey, where you have X is a biomass of a particular species, R the growth rate, and D is the interaction matrix, which is shown here. Okay, and you have a kind of a noise term here with a prefactor, which it was derived in a different paper a while ago. I'm not going to get into the details, but this is the stochastic model we use to reconstruct network. For example, this network, which is kind of a star-shaped, is represented by this kind of interaction matrix. So we actually increase, we can increase the Ds, DIAs, which will be a kind of a measure of the interaction strength between species. So different Ds, and I will show you how dynamic change as you increase the interaction strength, so we can see how the ability to detect the network actually changes. And the other term here is the diffusion term, where you have a gamma factor, which give you a possibility to control the intensity of the noise of the Brownian here. So so we tried to see how well different methods, again, correlation covariance, inverse covariance, and also an algorithm that has been developed to actually detect this kind of network perform in reconstructing a network that we know, and we put noise, and we generate time series of this we generate this time series, so we can infer. We generate this species by species kind of interactions of statistics, and we can infer. We use covariance versus covariance and another method which is called limits, and we can reconstruct a network. So the idea will be that if the method is good, we will output the same network that we were simulating. Unfortunately, as I will tell you, this rarely happens. So there is sort of a indeterminacy problem because most of the methods assume kind of a particular distribution, which is normality, and our data is far different from normal data. So that's one of the problems associated with this. But the other problems are a little bit more subtle and are related to the way that environmental stochasticity can generate covariance motifs. So this is what we did, and we actually try all these different trophic motifs with 4 species, and we try different methods. We simulate them, we generate time series, we apply all the 3 methods, and we test how many times particular methods provide a good insight in terms of the topology of the network. And here is kind of how the sensitivity of the true positive rate of the proportional way of number of times that you actually reconstruct a true link. You see that this tends to increase as the interaction strength actually increases for some network, but from some other network it does decrease. And and this here is the difference between the different levels of noise with this kind of factor here. So you see that the methods kind of perform with a large variability, but it depends on the kind of network that you are reconstructing and the kind of motif that you are trying. So some of them have high sensitivity in terms of this is the black bar is the number of true positive. So usually for some method like the covariance, you can get a higher than random. Is that the dash line is what you would expect as random reconstruction for these methods, but for some other network you are completely right on the random kind of reconstruction. So not very good. So so in general, for some network motifs, some methods are very good. They are very insensitive to noise level, by the way. And in terms of comparison of metrics, as I said, the different metrics perform differently, and they tend to change with the interaction strength, but not with the amount of noise. So that's noise doesn't affect really the sensitivity or the specificity. Remember, the sensitivity is the probability of actually doing the right thing or identifying a true positive, and the specificity is a true negative rate of identification. So so even though they are sensible to the interaction strength, the sensitivity is not great, and most of the time it's not different from the random one, which is 0.5. So that's what you would expect if you just do a random kind of a reconstruction of links. And part of the problem is associated with the fact that this time series have a long transients, and you can understand a little bit about that using analyzing the interaction matrix so what computing something which is called the reactivity, which is associated with the largest eigenvalue of the matrices, and you see that they have very some of them for higher sensitivity they achieve large values, and they tend to increase. So this means that they become increasingly transient, becoming increasingly large, and so they don't settle into the real equilibrium for a long time. So transients grow, and let me show you, this is kind of the same thing that just for different species on the eigenvector associated to the eigenvalue, but let me show you here some of what actually is going on. So this is a true network. Here is a larger experiment. This is for 100 components. Okay, so you have the trophic level here and here, and this is the kind of the average true network in terms of the correlations that you get, the true correlations for the true network as you simulate it. And this is the reconstructed one. You see that for D 0.01, you actually get something very random. And when you start increasing the interaction strength, you actually start seeing some structure. So you start kind of reconstructing a lot of significant interactions along this area here, and among high trophic level species, and among kind of a high trophic and low trophic level, and kind of there's sort of a pattern emerging just because of the level of interaction strengths that you put into the matrices. So in general, what is happening is that as you increase the interaction strength, so you start increasing the kind of reactivity of the system. Here are the probability density for the different reactivities for these matrices, and you see that as you increase the interaction strength, the reactivity or the time it takes for the system to go back or to reach equilibrium gets much larger. So what is actually happening was kind of predicted a long time ago. Back in 1988, Peter Jotsis, which is a famous theoretical ecologist, kind of wrote a paper saying, well, there is some indeterminacy of ecological interactions. And he was completely right. What his point was that if you do a perturbation experiment in a network, meaning if you start, he was talking about some particular experiments which are called press experiments. So you suppose that it's like you keep a pressure on a system, like you start putting pesticides on a system, or contaminating, or you put in pollutants in a system, or climate change is a press experiment too. So because you keep pushing, and he said very much as I kind of cut and paste here from his own words, said, for those doing practical work with environmental impact, it is of crucial importance to understand that short-term observation of environmental impacts that can be viewed as press perturbations are close to useless for estimating probable long-term impacts. So he's saying, if you want to understand what is going to happen when you actually press the system, you keep pressing the system, you will not be able to, because it's going to take a long time, a long transient, and it's going to be a sort of indeterminacy, because causality is going to propagate through the system in different ways that you are not going to be able to reconstruct. And he continues saying, moreover, as the present study makes clear, predicting those long-term effects not only requires data on the strength, the interaction strengths of many interactions in the system, it requires very accurate data on many interaction strengths. This is a daunting prospect indeed. So, and our conclusion is positive on the negative side. So we make with these two kind of analysis, we make clear that reconstructing ecological networks is a very complicated kind of endeavor. It's an open challenge in many ways, and we have to devise new methodologies to be able to understand how we can approach this this kind of complexity. The ones that are available so far are not very good, and most of the time, as I show in the second part of my talk, they do not do better as just performing random kind of random reconstructions of links in the networks. So I have a couple of conclusions. So environmental and settlement preference may outweigh biotic interactions. If you use kind of co-occurrence data, it's more important the place than the interactions. And some species, particularly habitat engineering species and non-trophic positive interactions, leave a more detectable signal than other interactions types. Also, co-occurrence network are available for analysis of community dynamics as an epiphenomenon combining interaction and environment, rather than simply as a result of pairwise interaction. So you will always have kind of an environmental. In this case, it's not environmental stochasticity, but we are talking about the real impact of the environment in terms of species living in some particular places because they can persist there, or because of how we call it niche related reasons. And also say that whole network of species interactions are detectable sometimes by covariance and correlations, but increasing stochasticity doesn't have too much of an impact, and no random selection of network motifs is increased as you increase also the interaction strength. So that's what I had prepared, and I would like to thank Sergio Navarrete, who is coming next. So he will explain my talk. Thank you very much.
Thank you, Pablo. So maybe there are questions concerning your talk.
I was in time, isn't it?
In fact, you were a bit fast because, as far as I understood, you were
until the first slide.
No, it's still 30 minutes more, I think.
Oh, good.
So I don't know what we do, but so
there's a talk. Can you
because
say
in fact, there are questions. Oh, sorry.
Yeah, there are some questions here.
Yes. Okay. Is there a question between what you do and matrix reconstruction?
Is that the whole question?
I think yes. In fact, matrix reconstruction is a hot topic in statistical learning.
All right. Yeah, I think it's related. Some people is trying to use machine learning and all sort of deep learning algorithm to reconstruct this. But I don't know how the connection really is, but there is one. And also in terms of trying to infer causality. In fact, we are doing some work now with people like INRIA in trying to using some of the big large data sets on microbes on the ocean. There's probably the Tara expedition is a couple of French consortia which actually are they have this expedition going across the oceans. They have done already a couple of them, and they collect at each sample. They collect at each site. They collect samples of 3 different depths, and they take pictures of organisms of phytoplankton, and they take the genomes of the samples, and they analyze all the species of bacteria and microorganisms present, and they have this huge, amazing meta genomics sample. They have published a lot of interesting papers. So we are trying to infer, trying to use that database to infer some interactions. But in this case, if you have the gene, so you can actually try to go and annotate this gene and try to know what kind of function is this gene, or what kind of protein is this gene codifying for, and you can try to put that product into the metabolic network. And there, it might be easier to reconstruct the interaction, because you will have also physical chemical constraints, and you know a lot about there is a lot of knowledge on the metabolism of different kind of organisms. So that might be that's something that we want to use artificial intelligence techniques and this kind of reconstructing this metabolic stuff. But I have no much details, but yeah, it's important.
Thank you, Pablo. So I don't know if there are other questions or not. If there are no other questions, I'm sorry.
Oh, he said that have you used the bootstrap? Yeah. No, we haven't gone that where using that, but it might be an interesting suggestion actually. So we can talk more about that if you like. Enough, yeah. You're welcome. Yeah, to treat the dependence on the time series. Yep, that's good. Thank you.
This is in fact something very complicated to bootstrap time series, and that's also something of a special interest, because you cannot bootstrap it term by term, but you have really to use some kinds of block bootstraps techniques. That makes the thing quite theoretically quite complicated, and the right way to do that is often by using some renewal properties of processes, and so you can just use independence of some parts of the chains in order to make to resample. That's either that or making this in a more approximate way. So okay, so I think that there are, so this means at least that there are lots of topics and we to consider, and this means that we have like 4 years to work on it, and together with Victor and all the people in this conference, it will be a real pleasure for me. And so I suggest, in any case, that we now just stop and we come back with Sergio at within exactly 27 minutes. That means
wow, a big kind of break.
Yes,
awesome.
Because that's in fact your talk was planned
short
until 4:45. In fact, I discussed with your co-author and he suggested this time, so we planned it. And so
perfect.
Let's. I think it's more convenient if we come back with Sergio later. I wanted to thank you personally for your very nice talk and also
thank
for
you,
the
Paul.
very beautiful photos which illustrate that
of real things.
Yes, exactly. Thank you very much. So
yeah, thank you, Paul.
Let's stop for a while and so we put a pause on the conference until the precise normal time. Okay?
Okay, thank you. Bye-bye. See you in a while.